Okay, so good morning, everyone. Let us have one more session on this antifungal agents. So, in the last session, we have revised what are the antifungal agents, what are the fungal pathogens, and what are the kinds of pathogenic disorders these antifungal agents or pathogenic conditions which these kinds of fungus can create. Okay, so there are so many types of funguses: systematic mycosis, subcutaneous mycosis, cutaneous mycosis, and superficial mycosis. So this classification of mycos, or we can classification of this fungus. So that is based upon this uh, infections where they can cause, means whether it is cutaneous, which is or subcutaneous, so superficial or systematic. Okay, so depending upon this, these all funguses have been classified. Then after this, we have seen so many other kinds of pathogenic disorders, which may be this oral candidiasis, or it may be cutaneous infections, so which can cause the several kinds of cutaneous, subcutaneous, this fungal infections. Okay, so ringworm, tinea capitis. So these are the kind of fungal infections which can occur. So classification of antifungal agents. So this classification of antifungal agent is based upon the way or the synthesis. Some of them are synthetic antifungal agents and some of them are antibiotics. So synthetic antifungal agents, we have seen acid and acid derivatives. Majorly, these acid and acid derivatives are fatty acids, benzoic acid, and salicylic acids. So there are the structures of these fatty acids is there on your screen. Benzoic acid and salicylic acid are the kind of non-fatty acids which possess this kind of uh, antifungal activity. Propionic acid, caprylic acid, and undecylenic acid. So they are fatty acids. Propionic acid and caprylic acid are saturated fatty acids, while undecylenic acid, that is obtained by destructive distillation of castor oil. So that fatty acid is basically unsaturated, whereas these two fatty acids are the saturated kind of examples. Triacid, so this is triester molecule, which is also possess this antifungal activity. Pyrimidine derivatives were generally 5-fluorocytosine. So this is the agent which shows this kind of one activity, antifungal activity. This is all not only antifungal agent, but it is also used as anti-cancer agents in some kind of infections where um, it is also used in as antifungal agent in combination with amphotericin B. So amphotericin in combination with amphotericin B, it is used as antifungal agents because amphotericin B will cause the perforations in the cell wall of fungi, and this uh, 5-fluorocytosine will get easily entered into the interior of the fungus cells. Okay, so friends, then another this mode of action. So 5-fluorocytosine that is metabolized through some enzymatic processes, that is selective deamination via cytosine deaminase enzyme in the to 5-amino fluorouracil. And that 5-amino fluorouracil, which is incorporated into the RNA, and that is basically inhibiting this RNA synthesis. So 5-fluorouracil that is getting converted into from 5-fluorocytosine that is getting converted into uridine monophosphate, diphosphate, and triphosphate. So kinase is an enzyme which is involved in phosphorylation of this, and it can cause this type of phospho. So that triphosphoderivative, I mean to say triphosphate derivative, is the key analog which can cause this type of, I mean to say, basically in this antifungal activity through the inhibition of synthesis of RNA. Okay, then we also studied these phenolic derivatives. So these phenolic derivatives which we have studied. So they also possess antifungal activity depending upon the this denaturation of the DNA is also I mean to say protein that is for this phenolic compound. So 5-parachlorometa methoxyl and cleophenol or chlophenol. So this is basically 5-chloro 8-hydroxy 7-iodoquinoline, which is competing with the coenzymes for metal binding sites. And it can be through the several oxygen flask followed by it can they be acid through some called Hart's method, because as you know that for halogen estimation this hard method is one of the essential method which you might have learned in your some of the inorganic chemistry practical during your first year foundry. Then it comes to thiocarbonate. So tall naphthalate is this kind of compound which is thiocarbonate derivative which is having thioester as well as thiamide. So that's why it is called tall naphthalate thiocarbonate derivative. So there is orthonaphthyl and metadolyl derivative is a kind of derivative that is essential that is also possessing antifungal activity. Under the synthetic class of antifungal agent, one more class that is azole derivatives. Basically, OLE that stands for 5-membered ring, and this I mean to say uh, clotrimazole is a kind of that another agent which is responsible for antifungal activity, and that is also present in each guard. The way clotrimazole is a API, active pharmaceutical ingredient of this each guard. So that is all used in some of the uh, fungal infections. Okay, and that is used for several topical infections like tinea, mucocutaneous candidiasis, vaginal candidiasis, and some of the systematic infections where it can cause severe GI disturbance. So that is also used in such cases. Iconazole, another kind of imidazole derivative, which is having the structure like this, IUPAC name is also there. And another substance that is ketoconazole. So ketoconazole is a substance having this imidazole ring and this piperazine ring in its 2D structure or in its chemical structure, and that is useful in some of the fungal infections like this. But it can cause hepatotoxicity, which is one of the major side effect of this drug. Then this Shankar Bhagwan Trishul, that is triazole derivative, fluconazole. It is having as shape of this molecule is like a Trishul of Shankar Bhagwan, Lord Shankar. Okay, so triazole derivative, fluconazole, uh, from I think one more, and that is responsible for inhibition of 14 alpha DB. So I have that chart with me. So this is choline, which is a kind of basically acyclic compound and having more than one double bonds. Okay, so scaline is acyclic one, and that is getting in the presence of some of the enzyme. It will get converted into lanosterol, then zymosterol, ficosterol, episterol, and finally ergosterol. So we the people require cholesterol. Cholesterol is active constituent for synthesis of steroids. Even our hormones, sexual hormones, especially testosterone in male and progesterone and estrogen in female. So these sexual hormones are also synthesized from cholesterol. Cholesterol is basically synthesized from scalene. Scalene epoxidase, 14 alpha D methylase, lanosterol, 14 D alpha methylase. They are some of the enzymes. But in fungi, in fungus cells, this is not cholesterol. Cholesterol is not that much required. They require ergosterol, and ergosterol is present in their cell walls. So that's why this is a key component for survival of fungus cells, or we can call any type of fungus. Okay, so coming to that, this our drug fluconazole is responsible for inhibition of this lanosterol 14 alpha D methylase. Thus, it inhibits ergosterol biosynthesis that is responsible for causing the potassium loss as well as some of the amino acids. And this one, allylamine derivative, where we are we have not seen some of this, but allylamine, where this CH2 double bond CH2, which is basically uh, ethinyl bond and which is connected with CH2, and that CH2 is connected with N, which amine. So this is a component called as allylamine. So allylamine derivative, that is basically this terbinafine, which possesses of tertiary butyl, naphthyl, and amine component. This is the IUPAC name of the same, where the double bond possesses E geometry. The reason is that both the hydrogens are on opposite side across each other, or else if we give the priority, this will be priority number 1, this will be priority, hydrogen will be priority number 2. And for this carbon, hydrogen priority will come over here, and this carbon will be another. So there are two ways, two priorities, one, one. So both the one priorities are on opposite side. That's why this allylamine का जो ये single double bond है ना, that possesses E type of geometry. The nitrogen of this allylamine that is connected with trimethyl, that is connected with N66 trimethyl, means the nitrogen either P है और इधर भी है, and that heptylamine का जो है that is attached with methyl, here it is, as well as naphthalene, and that is methyl group here. So that is connected with this way with methyl, naphthalene, methyl and naphthalene, and trimethyl. This is basically heptide, heptatuene. 1, 2, 3, 4, 5, 6, and 7. So 7 carbon containing, this is a big long chain, which is ultimately possessing at second position there is a double bond. At fourth position there is a triple bond. So ये जो YNE है ना, that YNE stands for our alkyl. So that is present at the fourth position. First position is connected with amine, so that they want to save from this. नाइट्रोजन पे भी मिथाइल है, एन का मतलब है नाइट्रोजन पे मिथाइल है, और 6 नंबर मतलब दिस इज 6 नंबर, वहां पर भी 2 मिथाइल, सो देयर आर टोटली 3 मिथाइल. दिस इज द वे दैट IUPAC name of terbinafine is basically given. This terbinafine hydrochloride is a synthetic allylamine antifungal agent, which we can synthesize in the lab. That is a kind of synthetic antifungal agent. It is highly lipophilic in nature, which tends to accumulate in skin, nails, as well as adipose tissue. And that prevents the conversion of this lanosterol to ergosterol by inhibiting the enzyme, what is called 14D alpha methylase. So by inhibition of 14D alpha methylase, this synthesis of ergosterol is inhibited in fungus cells, because we don't require ergosterol, we require cholesterol. But फिर भी थोड़ा pathway similarity है. It may be shikimic acid pathway or some other pathway. So that's why this kind of some side effects may be observed. Okay. In some of the conditions such as jock itch or tinea because of tinea cruris, athlete's foot, tinea pedis, and other type of ringworm infections, this fungal agent terbinafine can be used. Till the time we have studied all the antifungal agents which are synthetic in nature, which we can synthesize in our lab. Chemistry lab where we can go and we can synthesize the compounds. That's why they are termed as synthetic. Now we are going to change from synthetic antifungal agents to antibiotics. Now let me tell you what are these antibiotics. Already will be taught by some other teacher, has been taught by some other teacher. Antibiotics are the substances which are produced by microorganisms. Not neither yourself or myself. They are produced by microorganisms. Microorganisms can produce this substance. So what we are doing, we are on an agar plate medium, we are cultivating these microorganisms. By their cultivation, by a culture medium, once we have these microorganisms, then we can get antibiotics. We can deal with them. We can synthesize. I mean to say, we can manufacture them. इसी को बोलते हैं antibiotics. This is a difference between synthetic antibacterial agents or antifungal agents and antibiotic antifungal drugs. Alexander Fleming ने जो डिस्कवरी किया था पेनिसिलिन का, that is antibiotic. But yes, now today we can synthesize antibiotic in our organic chemistry labs. Synthetic protocols, synthetic steps are available to synthesize penicillin, beta-lactam antibiotic. So फिर भी that is obtained, that we synthesize, although we can synthesize in the lab, but it is also synthesized by the Penicillium notatum, Penicillium chrysogenum. So it is antibiotic. पर फिर आप पेनिसिलिन का कोई डेरिवेटिव बनाते हो सब्स्टिट्यूशन करके, क्लोरो के बदले मिथाइल, मिथाइल के बदले इथाइल, इथाइल के बदले समथिंग डिफरेंट. 4 मेंबर बीटा लैक्टाम के बदले आप 5 मेंबर सिफेमरिन बना देते हो, या फिर सिफालोस्पोरिन बना देते हो, या फिर ऑक्सोपिनाम बना देते हो. ऑल दीज आर डेरिवेटिव्स. एक बार आपको प्रोसेस पता लग जाता है ना, तो वी कैन मॉडिफाई इट, उसमें चेंजेस कर सकते हैं. दैट चेंजेस वी कैन अडॉप्ट फॉर द पर्पस ऑफ आवर सिंथेसिस. That way they are antifungal agents, and these are the antibiotics which are produced by the, whether it is by bacteria or fungus cells or some other. So that we will study in this in the coming slides. That antifungal agents, and one of the antifungal agent which is non-polyene antibiotic. Poly means more than one and ene stands for double bonds. So non-polyene means उनमें कोई भी एक से ज्यादा double bond नहीं होते. Even griseofulvin, if you look at the structure, I mean to say here there is no carbon-carbon double bond. Although यहाँ पे एक है, but it is a part of ring. There is no carbon-carbon double bond outside of the ring. So that is basically antifungal antibiotic, griseofulvin. Okay, so this griseofulvin that is obtained from Penicillium griseofulvum. Penicillium genus से ही वो बना हुआ है. And this is the IUPAC name of this. Benzofuran, this is benzo and furan. 2,1 cyclohexene 2-ene. So at this position, both the cyclohexene 2-ene is there. 3-4- dione. So this is 3-3 position. This is 4- dione. And then chloro is here. 2,4- 2-4,6 trimethoxy here. And then 6- methyl. So here is a methyl in this gisiofulvene, which is obtained from, as it is mentioned over here, Penicillium gisiofulvene. And it is obtained from Penicillium bacterium, Penicillium bacteria. That's why it's antibiotic. That can be used in systematic fungal infections of body, nail, hair, and feet. Okay, so it is going to arrest the cell division at the metaphase, and that can show the activity. And basically, it is benzofuranon derivative, and which is going to bind with microtubules. Once it will bind with these microtubules, then it will be able to show its anti-fungal activity. Okay. Now, after this Penicillium griseo another substance that is uh, polyene antibiotic, that is amphotericin B. So we let us revise this, what is called amphotericin B or polyene antibiotics. So before we go to the structure, we need to understand this. Penicillium griseofulvum will give griseofulvine, which is non-polyene antibiotic. But this polyene fungal antibiotic, basically they are having conjugated system of dynes. You have read the organic chemistry in your organic chemistry first year pharmacy program, where you might have seen there are many types of dynes, conjugated dynes, where two double bonds are separated with each other through a carbon dioxide. Two double bonds are separate, that is called conjugated. If two double bonds are in consecutive manner with each other, so it is called as cumulated dyne. If two double bonds are separated with more than one single bonds, so it is called isolated dynes. So isolated dynes, conjugated dynes, and cumulated dynes are the three types of the dynes. Compounds which are having two carbon-carbon bonds. So now friends, let us go to this, which is 26-membered polyene, or examples are natamycin. Natamycin is an antibiotic which is having 26 carbon ring chain. Organic chemistry, it is much difficult to synthesize a cyclic ring which is having 26 atoms. Benzene में तो 6 है, 5 and 1, only 6. एम्फोटेरिसिन बी का जो साइकिल है उसमें 26 कार्बन है, या फिर 26 एटम है. वो एक मूवी आता है जिसमें बोलते हैं 26 26 26, और उसके बाद कोरेक जाता है तो बोलता है 27. आई डोंट रिमेम्बर द नेम ऑफ दैट मूवी. बट ये जो आपका 26 है ना, वो नाटामाइसिन एंटीबायोटिक में है. देन another antibiotic, that is 38-membered ring polyene antibiotic. Uska ring site, I will show the structure to you, amphotericin B. Once you look at the structure, you will find it is a huge macrocyclic ring. Macro means bada. Okay, so amphotericin B and nystatin, candicin, they are the antibiotics. which possess 38, उसमें तो 26 और इससे भी वो 26 से भी आगे 38 membered antibodies. Amphotericin B. Look at the structure and go and count this 1, 2, 3, 4, and total 38 atoms are there. Then more active amphotericin E. Sorry, amphotericin B is more active than amphotericin E. Where amphotericin may, there is a carboxylic acid functional group. You can see how many double bonds? 1, 2, 3, 4, 5, 6, 7 double bonds. So it is heptaine. यहाँ पे कहीं पे लिखा था ना heptaine antibiotic. पहले होगा. So it possess total 7 double bonds. And it is carboxylic group which is attached to the macrolide group as well. But the side effect of this, it can cause nephrotoxicity. So many kinds of fungal infections due to candida albicans, etc., etc. But the one more good thing about amphotericin B is that this is a drug which possess, I mean to say, it was discovered in 1956 by a scientist whose name is Gold et al. So there are two antibiotics of this, which is obtained from extracts of Streptomyces venezuela. Streptomyces venezuela is the biological source from which amphotericin B is obtained. And there are two things, amphotericin A and amphotericin B. इसी जो स्ट्रेप्टोमाइसिस वेनेजुएला के जो उसका जो एक्सट्रैक्ट है उससे दो एंटीबायोटिक मिलते हैं, एम्फोटेरिसिन ए, एम्फोटेरिसिन बी. But amphotericin B is having better antifungal activity than amphotericin A. इसीलिए medicinally antifungal amphotericin B is used. And it is used in systematic फंगल इन्फेक्शन. सिस्टमेटिक जो अभी तक हमने देखा ना उसको क्रीम और उसमें अवेलेबल है. दिस इज गिवन फॉर सिस्टमेटिक फंगल इन्फेक्शंस. And also for leishmaniasis, which is called kala azar. In that case, amphotericin B is used. Nystatin, again, this is a 38 member ring antibiotic and having 1, 2, 3, 4, 1, 2, 3, 4, 5, and 6. So it is hexaene, having 6 carbon-carbon double bonds. So friends, here they have classified that it is a hydrophilic character, all COOH, hydroxyl group, etc., etc. Philic character, because they can make hydrogen bonds. And this is a hydrophobic character. इसला जो amphotericin का structure था ना, where all the bonds were double bonds. But here, only one double bond is saturated. Although it is same structure, यहाँ पे वही structure है, but it is hexyl in that possess this antifungal. Candidacine antibiotic, that is also a polyene antibiotic. Polyene का मतलब है कि more than one double bonds. So here 1, 2, 3, 4, 5, 6, 7, 8 double bonds are present in candidacine, which is used in some of the vaginal tablets for vaginal infections. Natamycin, which is a 26-membered lactone ring, and that is showing 5% of thalamic suspension, etc. So if it is not how many 1, 2, 3, 4, and 5 pentaene. If we ask, all of them are polyene, but if we ask hexyne, then this natamycin. If we say, sorry, this one nystatin, If we say uh, pentine, then it will come to natamycin. If we say heptine, then it will come to amphotericin. And if we say this hexine, then it will come to instead octine, then it is candidicin. So this way, there are several antifungal agents, and all of these antifungal agents have common mode of the action that they cause disturbance in this potassium level, hyperpolarization of fungus cells, right? I'm not sure whether Dr. Mehul Chorawala has taught you the cardiac cycle where hyperpolarization or something other, or some other teacher might have taught you this. So amphotericin is responsible for opening of the fungal cell wall membrane, in which will allow the potassium ions to come out into the cytosol, and that will cause hyperpolarization. And this hyperpolarization of fungus cell wall is a major cause for this killing of fungus cells. In that way, it can show either fungistatic or fungicidal antifungal activity. Okay, so basically, this 38-membered ring nystatin natamycin is 26-membered ring, but the side effect of nystatin is that it possesses the bad taste in mouth. Somehow, easily people used to call it natamycin, this medicine, as swish and swallow drug. Swish and swallow means chuskilo or galija. Natamycin is this type of. Okay, so let us stop up to this because we have majorly covered our antifungal discussion. So hopefully I shall may be able to explain you what are the antifungal agents, etc. Thank you everyone. Thank you so much.